Welcome back to the hands-on part of the metagenomics bin evaluation lesson. We will be talking through the CheckM software and see what its capabilities are and what kind of types of analysis you can do with CheckM. For this, we have a binder that is over here. It's available at github.com/biovcnet/bvcn-binder-checkm. If you type that, or you see it here at the top of the screen, that will bring you to this GitHub repository, and this has this launch binder button. Now, if you click, in addition, the README has a walkthrough of some of the commands that one can run in CheckM, as well as the steps to start from a publicly available data set. We will walk through those here in this video. I think we will not run the CheckM taxonomy workflow command, as is over here, because that takes about 25 minutes, but you can, without any trouble, run that by yourself. So, if you click Launch Binder, it will open the binder in the same page. So usually I click Open Link in a new tab and open it that way. It will take a little bit of time, especially the first time you load it. But what you should see after it's loaded is the following screen. So this is, you may recognize, the same directory structure that we have over here in the GitHub repository, now just in its own little, well, binderized bubble. And we can use the tools available here to open different tabs and visualize things in the binder. Now, if we go back to the tutorial here, what we are doing today is we're taking the completeness of genomes from this paper over here. This is a paper from my own PhD thesis where we extracted 23 genomes from a wastewater treatment metagenome. And the two reasons that I think this is a good idea is that it's a relatively small set, so you can run the various commands in CheckM relatively quickly. And the paper itself, because of when I did the analysis, did not use CheckM yet. So it allows you to compare the completeness estimate based on the gene set of DuPont et al. In 2012 with the gene sets that CheckM uses. So as part of this, let's briefly talk through how we would get the data here. So these steps are already done in the binder up until organization, but we'll walk through them here briefly. So the first thing you'd have to do if you want to look at data from a paper is you'd have to find the relevant accession number. Now this would be the bio project. Bio project number is a way of NCBI to organize all of their things together. And we see here that there is 23 whole genome shotgun master records in this, and these correspond to the 23 bins that we put, or that we extracted in that paper. What you can see is that if we scroll down here, we see a download link. This has definitely burnt me in the past. This does not download the assembly. This just downloads a text version, TSV version of this table over here. It's useful too. And we have downloaded that table, and it is this specific table here. So bio project number, assembly details.txt. And if we look at what that looks like, we can just click on it, and we see it's indeed just a tab delimited file with exactly that. Now, to actually get to the data from a paper, you go back up and you click assembly. If you click assembly, you get this big fat button at the top that says download assemblies. This will download a tar compressed file with these 23, what NCBI refers to as assemblies, but there are the 23 bins from the paper in it. And that is the point to which we've gone so far. So, and we see that this here is the tar file. So to analyze this, what we do is we go into terminal, and then into terminal, you'll see a terminal window open, and we'll type this. Now, probably will appear. Oh, there it is. So now what we've done is we've unpacked the tar file, genome assemblies genome fasta.tar, into NCBI-genomes-2020-0728. I downloaded it yesterday. What we now have to do, you see that each individual genome file is also still compressed. So we'll go into the, I guess we'll do that. We will go into the NCBI genomes directory, and initially in the tutorial set, we look at the README file. The README file is a relatively long file with all the possibilities that NCBI has. Now, one of the reason I wanted you to look at this file is you can download genomes from a single study, for example, by going through the website. You can also download these things in bulk from the NCBI genomes page. We'll not do this now, but it is useful to have as a resource and know that it is available for you. All right, so from there, where we'll go is we will unzip, oh, look at that, clearly I forgot that. We'll unzip the files in this directory. And then we'll type ls-lh, sorry, to just get a list view of all the files. And we now see that there's a number of files that are all genomic FNA files. So if I type head dash, well, head is fine actually. And just look at what this looks like. Oh, well, clearly that didn't work. We will see that this is a genomic FASTA file with in the header the information of this genome. Now, these are not super convenient to read, so we're going to change the bins to a slightly more convenient name. For that, we're going to take the assembly details file that we've just downloaded. The initial thing that we do is a little search and replace. That's what this little Perl line does. It replaces all the spaces by underscores, just to remove spaces from the file. And then what we will do, and this is not necessarily part of CheckM, but it is a quick and dirty way of renaming, renaming these files, is we do a little while loop. So we go through the assembly details table file that we have, and we make, and then this loop in the middle changes the name from the name representing the first column to the name representing the sixth column. And if we do that, and then again look at the contents of the file, we now see they're now named, contents of the folder, they're now named slightly different. So what we've done is we've changed them from a name starting with the assembly identifier to this taxonomy name, just because it's slightly easier to read, and it's also slightly easier to correlate with the other things that are available. So we now know that there's a few Bacteroidetes, there's a few Chlorobi, there's a few Chloroflexi, and a few things from other taxa. So that still not technically CheckM, but now we're going into the CheckM core functionality. We'll first have a look at how CheckM is organized. So the way it is organized currently is if you type the CheckM command, which is just CheckM -H for help, what we will get, we'll see that this is CheckM version 1.1.2. You'll see in the README that 1.1.2 has the issue that these plots do not work because of an incompatibility with Matplotlib. Donovan Parks, who is the author of CheckM, released an updated version recently. This is not yet available through Anaconda, which is why it's not in this binder. These here, this set of commands is the core, what I would call the core functionality of CheckM. So it's the set that takes your contigs, and your contigs are, as we've just seen, DNA sequences, not yet annotated, and identifies genes in them, picks those, picks the markers, puts them in a tree, and then picks a set that is appropriate for the position of each individual bin in the tree, and then takes the analysis for each individual, and then does a completeness analysis for each individual bin. This is summarized in what's called the lineage workflow. Now, we will not be able to run the lineage workflow within the binder because of memory issues. Placing bins in a tree is computationally very expensive. However, we will be able to run the taxonomy workflow in this binder. For the taxonomy workflow, we see that the we will see that that we have to specify where the for the taxonomy workflow, we have to specify where which taxonomic level we run this at. So for that, we have the taxon list command. The way CheckM commands are structured is we go, we type the CheckM core command, then we type the secondary command, and then we type the options that are available for the secondary command. So in this case, we're typing CheckM taxon list, then -H as in the README, and we see what the options for the CheckM taxon list command is. So for the CheckM taxon list, we have the taxonomic rank, which we want to show, and a temp tier. So in this case, we are going to look at the entire taxon list that is available. So CheckM taxon list. This takes a second. And then we'll see that there's a good number of well-studied species. So for example, if you have a Pseudomonas aeruginosa, it will actually, like, if you have a bin that is Pseudomonas aeruginosa, CheckM will use a set from Pseudomonas aeruginosa. Now we can go up from species to genus to family, and then also That doesn't let us go up further. Okay, we'll do this slightly different. We're going to do it with more. All right, change that in the, there we go. So now we'll see the top, change that in the tutorial. Now we see the top, and we see that we can scroll down, and we see there's a chunk of phyla available, but not all that many, all things considered. For example, there's only, say, 2, 3, 4 archaeal phyla, probably a little bit more. But there's also domain-specific genes. So if you don't know what kind of organisms you have in your bins, you can take prokaryote, which is the which looks at marker genes for archaea and bacteria. Or if you know that you have archaea or bacteria, you can look at either archaea or bacteria. Here you'll see the number of genomes that is available for that marker set, and then these are the genes that the markers have in common. CheckM uses marker genes and marker sets. The marker genes are the individual genes. The sets are the ones that are co-located in the same place, typically. So say the RNA polymerase has a number of subunits. Those subunits tend to all be in the same place in the genome. So CheckM calls those a marker set, which is why this number is lower than the number of genes. All right, so if we would run the taxonomy command, what would happen is we would run a CheckM taxonomy workflow. We'll first look at the options. So the CheckM taxonomy workflow has these options. It's basically, you type the CheckM command, then taxonomy workflow. And then the few key options there are extension. This is the extension of the FASTA files that your bins are in. In this case, we have FNA, so we'll use FNA. The number of threads, which is how many threads you want to run this with. The binder only has 1 available, but the example command still has 4 threads to illustrate the option. And then the rank, the obligatory arguments, the positional arguments are the taxonomic rank. In this case, we picked domain, as we see here. We picked bacteria because I know there's only bacteria in this data set. And then the last 2 arguments are the bin dir, so the directory where the bin is. In the example command, we put a dot. The dot represents the directory we're currently in. And then the output, in this case, you can name that anything you want. In this case, I named it CheckM out. I'm not going to run this right now. It takes about 25 minutes, and that would unnecessarily extend the length of this video, but you can use this and run it in the binder without problems. We did, however, also provide in the original, or in the parent directory, provided extra info directory. Extra info directory has a few things in it. It has table 1 from the paper, which shows us the single copy marker genes, the number of single copy marker genes that are present out of 111, and coding sequences, tRNAs and rRNAs. So you see that all of these have a decent amount of single copy marker genes and have rRNAs and tRNAs present. These few down here have a lower number of single copy marker genes. This is because they are organisms from the candidate phyla radiation. And the candidate phyla radiation has very reduced genomes, and a lot of the universal genes are not actually present, because you see that number of contigs for these are actually lower than for many of the others. The other thing that's present here, and this is very low resolution, is the plot I showed in the bin evaluation lesson presentation. So we have here is each column represents one of the 23 genomes, each row represents a gene. So this shows a visual representation of the completeness and contamination that is also present in this table 1. So we also have a list of CheckM lineage workflow results and the CheckM lineage workflow out. So we'll first have a look at what we're doing, or what we're looking at in this out directory. So the directory where the contents of the lineage workflow were written. So we're going into the extra info directory, and then we go into, and then we list the contents of this lineage workflow out. Now, there's 2 directories, bins and storage, and then there's checkm.log and lineage.ms. Checkm.log just contains the output that CheckM writes while it is running. So if we go into this directory, and do log, we see that it's has a timestamp and just goes through the command that we ran, and then starts initially with the first command, placing bins, reference genome tree, and so on and so on. Head just chose the first 10 lines of this file. Lineage.ms is the lineage file. And we see this is a rather opaque looking file. This shows for each genome, such as this one, it tells us which markers it has used. So it takes the, tells us what the marker lineage is, what the node in the tree is that it used, and then which markers that marker lineage corresponds to. So this is a file that is required for some of the other outputs of CheckM as well. In the taxonomy workflow, if you run it for yourself, the lineage.ms will not be called lineage.ms, it will be called bacteria.ms, because we've set a fixed set of marker genes for that one that corresponds to that lineage in bacteria. Now, the other thing that we're seeing is that there's a bins directory and a storage directory. The bins directory contains one other subdirectory for each of the bins, and that contains these 4 files. In the taxonomy workflow output, it contains 3 files. It doesn't contain this tree file because in the taxonomy workflow, there's no placement in tree. These files are the annotations on this, on each bin. So predicted genes in amino acid format and predicted genes in GFF format. And then this is HMM search output for each of these, for each of the genes that are predicted. So then the storage, oops, I keep forgetting, -OH. This shows, this has the files that are used to write the final CheckM output. And then here in tree, there is the files for each of the 43 marker genes that are used to do initial tree placement for each of the bins. So this one, for example, will contain 23, the 23 sequences that matched this specific HMM. And then here, this is a concatenated set of all those. Then CheckM AIQI actually is the one that has, this contains all of the duplicated genes that CheckM detects. So if we look at this one, we'll look at the Anamox organism. If we look at this, we see that there were 2 genes duplicated in Candidatus Brocadia sinica, that is the Anamox organism in this Anamox wastewater treatment plant. And if we look at our supplementary figure, we see this is column 1. There is 3 genes duplicated according to this figure, but 2 of those will actually be the same as the 2 that we see here. Now, if we look at what that looks like, yeah, that doesn't work that way. We see that, well, these are short genes. They're ribosomal proteins, or this one is a ribosomal protein. I don't know what this one is. I think it's a ribosomal protein. We see the sequences, and we see they're not identical of these two genes. And you can use these sequences to assess whether you think that this contamination is real contamination, or whether you think it is an actual gene duplication. One way to do the low throughput way to do this would be to just blast them. And there we go. For argument's sake, we will quickly blast these. Oh no, shit. Beep, beep, beep. All right. So we're showing results in a new window. So we can copy and paste the other one as well. Now, I don't recommend doing this with all bins, especially if you have many. But again, if you have bins that you're particularly interested in or spend a lot of time talking about in your paper, in your analysis, this type of work is worth doing. Now we see that it's not a ribosomal protein, it's a DNA J. So we see that this thing has 100% similarity to Brocadia sinica. That's right, because that is that's the actual organism that this is. We also see that this one here, okay, is duplicated. So this is the actual DNA J. We also see that the other one is duplicated in at least one other Anamox bacterium and fairly common in, oh no, there we go. These are all other Anamox bacteria as well. Fairly common in other Anamox bacteria as well. As you go down, you see that that here the first non-Anamox bacteria you hit are things like Desulfobacteriales and some things that are called Cryosericum, whereas here it's a different set, but also still DNA J. Okay, so beyond this, there is a decent amount of there's a decent amount of other options that CheckM has. So we'll look at CheckM's other capabilities again, and by typing CheckM-H, oops, and we'll see that beyond the top, what I would call core functionality of CheckM, there is these 2 sets of plotting possibilities, a set of bin exploration and modification, and a number of utility functions. We'll only have a look at the plotting functions, the general plotting functions, because some of the other things require either BUM files or the full assembly, or because I know there isn't any purpose for example, the unique command in this set. So the reference distribution plots, as I've mentioned before, unfortunately do not work in CheckM 1.1.2. They work in CheckM 1.1.3, but that's not yet available through Anaconda. That has something to do with updating, Matplotlib updating their standards and no longer being compatible with code in CheckM. So we'll use these three general plots. We'll start with the NX plot. And we're running this on the NX plot. We just require the bin information. It will complain at us quite a bit. Sad. Now, what's happening? That's because I'm not in the right place. Yep. Right, the NX plot. It will complain at us quite a bit because there is a deprecation warning in Matplotlib as well. But it will still write the plots. What an NX plot is it's a variation on the N50 metric. The N50 metric is the amount of contig, or the fraction of the length of the contig that, no, hold up, the fraction of contigs that combine to be more than 50% of the total genome. NX substitutes that 50 for an X because it is multiple different plots. So we've now written this to extra info plots, and we'll look at this Acidobacterium. We'll open it in a new browser because the tabs are not great. And you see here that this, for example, is not great. There is a couple of long contigs, but then by the time you get to 50% of the genome, the sequence length is the contigs that are in there's 50% of the genome is present in contigs 100,000 base pairs or longer. And then if we go further down, 90% of the genome, for example, is present in contigs about 20,000 base pairs or longer. So we can do, you can look at how, like these are the more traditional metrics of bin completeness. So we could look, for example, at this. Let's do candidate division WS6. Do the same thing. I think that should be. Yep, so there's a one, this is a very short genome, and there's one really long contig that is more than 50% of the genome. And so we see that immediately 50% of the genome is present on contigs that are 0.7 MB or longer. And these are, here you see that there's a few long contigs. All right. Oops, so, not intended. We can do the same for, so, oh, that's fine. Sadly, I've lost all my previous commands. We can do the same for histogram plots, and we'll see that it will now write length histogram plots for all this, for all the bins. And so we'll have a look at what those look like. Basically the same data visualized in a different way. So again, here you see sequence length in kilobase pairs and number of sequences out of the 65 that are that make up this set. So you see that the length distribution in this particular bin is decent. Again, we can look at that same WS6 bacterium, and we'll see a much more spread out view where we see there's actually no short contigs and just contigs that are much longer. Okay. Then the last thing that I still wanted to show also is the CheckM marker plot functionality. CheckM marker plot functionality has, as you can see, an extra argument. It has the, well, we'll actually look at that here. CheckM marker, no, sad. Plot dash H. All right. All of these have very specific settings that you can choose and play with. The one additional positional argument is the results directory. So the directory is specified using the QA command. So that is a directory that is run as part of the CheckM workflow. And in this case, we've called that extra info CheckM lineage workflow out. So we specified that as an additional command in this one. Otherwise, it's still the bin directory and the place where we're going to put the plots. So we'll run that too. And again, it will complain because of the version of CheckM, but it will, the one additional thing that this specific plot does is it shows you how well your marker genes are distributed across your contigs and which contigs in your bin are supported by marker genes. So that is an important thing to know, is where, and what that then looks like is these marker pause plots. And I'll take this one again, just because I can. Open in the new tab, and we'll see that these are the contigs. We'll see that for this specific Acetobacteria bacterium, it's estimated at 87% complete, 1.7% contamination. And the markers reside on 30 of the 65 contigs in this bin. So 35 of the contigs are not necessarily supported by this completeness estimate, but those 35 contigs also are only 25% of the 25% of the bin. But that said, as I showed in the presentation, that doesn't, that means that they're not supported by this completeness estimate. Now, these colors over here show, are shown here in the legend, show each block is, in this case, 1900 base pairs. And the color shows whether there's 0, 1, 2, 3, or 4 or more marker genes present within those 1900 base pairs. So you see that for this relatively short contig, there is a whole stretch that basically, oh, there we go. You see that there's a whole stretch that has 3 or 4, or in some case 2 marker genes present at the beginning of the contig. So you see that this is what I was getting at, that marker genes are not universally distributed across the genome. They're clumped in sometimes large groups over small areas of the genome. So it is important to keep that in mind. Now, this is still running and writing more marker genes, or marker gene plots. We will have a look at the output meanwhile. This is the actual CheckM results. The CheckM results look like this. They're not, there is a way of having a formatted CheckM result, which is less useful for importing in things like Excel or other programs, but easier to read in this format. But what the columns are in this tab delimited file is the bin ID, marker lineage, number of genomes, number of markers, number of marker sets, and then 1, 0, 1, 2, 3, 4, 5 plus completeness, contamination, and strain heterogeneity. So completeness, let me walk through what all these columns mean. Bin ID is the name of the file. So whatever you ran into it minus the extension. So in this case, our extension was .fna. So it takes the name of the file minus the extension. The marker lineage is the lineage that it uses to assess completeness. So for the Acetobacterium that we've just looked at a few plots of, it used the kingdom bacteria with the UID 3187. For this Armatimonadites bacterium, it used kingdom bacteria as well, with a smaller or with a different UID. This means that it's even though it's still at the kingdom level, it's a different node in the tree. And you see that the number of genomes included in that node is variable. So in this case, it's 2,258. In this case, it's 1924, even though the name is the same. Now, we have the numbers of markers and the number of marker sets. The number of markers here is 188. Number of marker sets is 117. So you expect the marker sets to always be less than the markers. The reason being that some of the markers co-locate. That's what's called a marker set. Then 0, 1, 2, 3, 4, 5 plus is how often markers are detected. For a good genome, you should see things in the 0, the 1, and the 2 range. 3, 4, and 5 basically should all be empty. So in this case, for this Acetobacterium, what we have is we have 188 markers. We see 15 are present at 0 times. 170 are present 1 time, and 3 are present 2 times. And then there's no gene that's present 3, 4, or 5 times. So that's good. That's good. This is called 87% complete based on that, and 1.7% contaminated. So 3 genes out of 188 is slightly less than 2, so 1.71%. And then strain heterogeneity is 0. I haven't talked about strain heterogeneity yet. The only what that means is whether the markers that are duplicated are very similar to one another. So what you're looking at is you align your markers, as we've seen in the AAI_QA directory, which we looked at in a bit, or which we looked at a bit ago. If those 2 genes are more than 90% similar, it will count as strain heterogeneity. I am not entirely convinced that terminology is sound, but it is the terminology that CheckM has chosen. So we see that, for example, here is a strain heterogeneity of 50%. There's been 6 genes that are present twice. So 3 of those 6 genes in this Nitrosomonas organism are more than 90% identical to one another. All right. So that's what the baseline output looks like for the taxonomy workflow that you can run within this binder. This lineage will always be the same. It will just be one fixed lineage, and you'll have and then that also means that markers and marker sets will always be the same number. But here you can, for example, see that Nitrosomonas is a gamma or beta proteobacterium, has a smaller number of genome that are included, which also means that you have a larger number of markers. So in this case, you use 400 markers instead of 188 or just 140. So that means that your accuracy will be a lot higher for the Nitrosomonas quality estimates. So what we see is that this gives a fairly nuanced view of what the completeness and contamination of these genomes are. That is comparable, I would say, to what we see here. Maybe in some cases slightly different. We'll see that the results here, 67, 65, are on the very low end, even though these are genomes that are in very few pieces and are relatively high quality. And this is because some lineages lack large quantities of marker genes. All right, I will leave it at that. Feel free to contact me if there's any questions about the binder or about running this. And yeah, happy bin evaluating.